Let's do an exercise.
Let's do an exercise.
Hey.
Hypothesis is a liability.
Mm.
I'm going to tell you about a data set that we're about to assemble.
Okay.
I'm not going to tell you what it's for.
Because you don't want to bias me.
I don't want to
wanna
bias you with
prime-
my hypothesis.
Yes,
right?
I need to be unprimed.
So what we want to do is, if you have a plant, right? So you have a plant, it has roots, right? So, those are the roots, very beautiful roots.
Mm.
And then it has some leaves,
Mm.
right? So let's have a second leaf.
Beautiful leaves.
Beautiful leaves, right? So
You're a plant guy.
yeah, I'm a plant guy now. I'm a proud plant researcher. Right, okay, so
Okay.
let's say that's a very simplified plant. It's not very
Okay.
beautiful, and we're going to grow two types of plants. So there's two types that we're interested in.
Okay.
There's the C3 plants,
Mm-hmm.
which is just like a standard, normal, boring plant.
Mm.
But there's also C4 plants, and they have a slightly different way of doing photosynthesis.
Mm.
So in C3 plants, all the photosynthesis happens in the same type of cell. In C4 plants, it's distributed between two cell types.
Division of labor.
Yeah, they pump CO2 into a separated cell, and then that's where the CO2 is converted to sugars. And that makes the process more efficient because you have a higher concentration, so the reactions happen
Did C4
more
evolve
effectively.
from C3?
Yes, C4 evolved from C3.
Like they were climbing Mount Fuji?
They were climbing Mount Fuji. You know my paper about C4 evolution. Exactly. So now we try to understand that better. What we do here is we take related C3 and C4 plants. So they look pretty similar, but one uses C3, one uses C4. And what we do is we measure the temperature at the leaves and the temperature in the roots. And we measure the amount of photosynthesis, right? We measure how much CO2 is converted to sugars per minute.
Mm-hmm.
So that's what we look at. Let me see.
Okay.
Do we?
No,
do
don't
we
don't
also-
give me the hypothesis. Though.
No. I'm not going to give you the hypothesis. I'm wondering if there's another important variable that we collect.
So by
No, not
measuring
really. That's it.
photosynthesis, you mean you measure CO2 like
Uptake.
uptake?
Right. So
That's photosynthesis.
yeah, that's so it tells you how much CO2 was converted.
So
to
you have a measure
something.
of photosynthesis, you have a measure of temperature at both the stem and the leaf,
the root and the leaf,
the root and the leaf. And for every plant, you also know if it's C3 or C4.
and we have some replicates.
And so you have this data. And is the data a time course? No.
It can be a time course, yes.
So would you say that the data is over time measuring the temperature at the root, the temperature at the leaf, the photosynthesis rate,
Mm-hmm. Yes.
and whether it's C3 or C4? This is the data.
Yes.
Is that right?
Yeah.
Okay. Now, let's say I have this data. What would I do with it? But I think I would, like a poet, compare everything to everything else. I think I would ask if there are different conditions. I mean, you know what I would do is PCA on the data, actually. Where, because each thing here is a measurement, right? There's a measurement.
Yes. But PCA, you only have really 3 variables for
Yeah.
2 different types.
No,
So-
it's actually,
Does it make sense to do PCA on 3 variables?
it's like 4 variables. It's 4. I'm going to give it all 4.
Okay.
And then I'm wondering what you can see from it, because it could be that when I do it, these are the data points, like the columns, right? And then I might see the things diverge, like this is the C3 and intermixed here is also C4. I might see that, and then I might ask, oh, well, what is separating this? And it might be that different temperature or different phosphorylation, different synthesis. I had a block with phosphorylation,
Yeah.
photosynthesis.
Oh, okay. That's why you
You're like, what?
phosphorylation. Yes.
And so I think I would just let the data do the talking. Actually, that's a good advantage of principal compliance. Just look at what is PC1. See here, PC2 distinguishes C3 from C4. PC1 seems to capture, I mean, what if that's temperature? So I think by PCA, I could tease apart the different factors. That's the one thing I would do. But that's not a hypothesis. That's just me studying the data.
No, of course, but that's totally fine. So that way, essentially, that would be a way to say what distinguishes these two types of plants. Is it more temperatures or is it more photosynthesis that distinguishes them, or a combination of the two?
Right.
Okay. Yeah, that's interesting. That's cool. Anything else we can do?
Not really. I think I would also, I mean, it seems clear, you haven't said it, but it seems clear to me that you're looking for a difference between C3 and C4. Like that seems to be the, what do you call it?
Yeah, of course.
So you just
no, but I'm not telling you what difference I'm expecting.
Oh, you're not telling what you're expecting. That's good. So I'm actually much more unbiased. So you're obviously looking for a difference. What would it be? Well, it could be that C4 insist on a difference in temperatures while C3s don't. Because the way you've described C3 to me, it's like cell autonomous, and C4 is more like cell non-autonomous. Autonomous means you can just focus on one cell and capture everything.
Yeah.
And with C4, if you focus on one cell, you don't capture everything.
Yeah, that's right.
So I guess you're asking like what conditions need to be met. But the root is so far away from the leaf, and I thought C4 requires cells to be right next to each other. Is that not true?
Oh yeah, but the division of labor is between two types of cells
that are neighboring
in the leaf.
So what does it matter what the root is? Oh, that's
that's
for you to know and for me to not be biased about. So, well, what could be? I don't know what you were thinking there.
You don't need to guess, right? The question is there anything else?
Is there anything else?
Data?
Yeah. I mean,
I think this is a difficult data set for this exercise because it's easier if there's more dimensions.
Yeah, there's not too many dimensions here.
Yeah.
And I still can't figure it out, let it be said.
No, but
the goal is not
for me. The hypothesis is quite original, right?
Oh, what is the hypothesis?
Well, the hypothesis is that in C3 plants, they cool by evaporating water, right? So they take up water through the roots and then they evaporate it, and that's how they cool. That's how they control temperature. And our hypothesis is that in C4, that's partly true, but they have a second way of cooling, which is circulating water between the leaf and the root. So that heat is actually transferred to the soil.
To dissipate there.
Yeah, to dissipate to the soil rather than just dissipate to the air. And in that way, they're more efficient in how they use water.
Oh, so these C4s are not even just cell-to-cell communication, but entire organism communication.
That's what we think, right? So we think that actually the roots will heat up.
That's
in
very
C4,
cool.
But not in C3.
That's very cool. Well, thank you so much for explaining.
Yeah,
You're
welcome.
I hope I was of service.
Yeah, no,
this
is fun.
Would it kill you to explore the?
No, that's certainly something we'll do.
Kill you.
No. Okay.